Hi friends, we'll start with the next chapter, cell biology. In the previous video we have discussed about biochemistry part B questions. Now we will start with cell biology. So the next question is, what phenotype would you predict for a mutant mouse lacking one of the genes required for site specific recombination in lymphocytes? Okay, so first we will study a little about site specific recombination. So let's begin. In site specific recombination process, there is the VDJ joining, right? So we'll start with site specific recombination. And here what we see that here there is the involvement of VDJ joining. So what we see here in this process, which is during B cell development, and here V and J segments for light chain and VDJ segments for heavy chains are joined together. Okay, for heavy chains are joined together. Okay, VDJ are joined together in case of heavy chain and V and J are joined together for light chain, right? To form what? I write it here, are joined together to form what? This is going to form VL and this is going to form VH region. VL coding sequence and this is going to form VH coding sequence. Okay, and this process of joining, it is mediated by VDJ recombinase. VDJ recombinase. Here we write D in bracket. Okay, and this VDJ recombinase, it consists of two parts. What are the two parts? The first part is lymphocyte specific enzyme. The first part is lymphocytes specific enzyme, and the second part the enzymes which help in DNA repair. Okay, first part is lymphocytes specific enzyme and the second is the enzymes which help in DNA repair. Now this lymphocyte specific enzymes, okay, this lymphocyte specific proteins of this VDJ recombinase, okay, this lymphocyte specific proteins, this lymphocyte specific proteins which is catalyzed by this enzyme, they are encoded by two linked genes. Okay, this proteins, they are encoded by two linked genes which are RAG1 and RAG2. Okay, they are encode, so we can write this RAG1 and RAG2 encodes lymphocyte specific proteins, right? And what does this RAG stands for? RAG stands for recombination activating genes. Okay, and this is catalyzed by lymphocyte recombinase. This we have discussed, lymphocyte specific enzyme, right? Lymphocyte specific enzyme will catalyze the formation of lymphocyte specific proteins. Okay, next we come to that these RAG proteins, what is the role of these RAG proteins? These RAG proteins, we write the proteins in capital. These RAG proteins, what they will do, they will introduce a nick. Okay, they'll introduce a nick double stranded break. They will introduce a double stranded break at the flanking DNA sequence. Okay, what's the role of these rag protein? And after this double stranded break at the flanking DNA, what happens? It is followed by rejoining process. And this rejoining process is mediated by both the rag proteins, that is rag1 and rag2. Okay, and also by some DNA double stranded repair enzymes. Okay, so those individuals who are deficient in either RAG gene, any of the RAG gene, in that case, the mouse becomes immunodeficient. So if any RAG gene is deficient in mouse, what is the phenotype? The mouse will become immunodeficient. Why? Because now they are not able to VDJ joining and they are very susceptible to infection because they are unable to VDJ joining. Okay, as a result of which these kind of mouse will not have the functional B and T lymphocyte. As a result, these mouse are immunodeficient. So I again recall what we have learned. So first of all, what is site specific recombination? Here a process is involved which is VDJ joining. In case of VDJ joining, the VJ joins for light chain and VDJ join for heavy chain. Okay, they form the VL coding sequence when it is the light chain and VH coding sequence when it is the heavy chain, and this is mediated by enzyme VDJ recombinase. And this enzyme has two components. One is lymphocyte specific enzyme, second is enzyme which is helping DNA repair. Correct. Second is this lymphocyte specific proteins, they are encoded by rag1 and rag2 gene. Okay, here the enzyme which act as lymphocyte specific enzyme, which is number 1. Understood? First component. Now these rag proteins, what's the how the process goes on? These rag protein, it will first introduce a double stranded break at the flanking DNA. After the break, there is the joining process, and in the joining process both the rag proteins are involved. Okay, as well as the enzymes for the double stranded DNA repair are involved. So this is the second component of the enzyme. If you are understanding, first is the lymphocyte specific protein, second is the second component of the enzyme which is involved in the repair process. Now coming to our question, if any of this RAG gene is deficient, any of this gene which is required for site specific recombination is deficient in mouse, the mouse is not able to show VDJ joining. In that case, the mouse becomes immunodeficient. It cannot develop the functional B and T cells. Got it? So answer will be 2. That is, the mouse will become immunodeficient. Moving to the next question. Question number 26. The key determinant of the plane of cytokinesis in mammalian cells. So please remember, sometimes the question is asked for plant cell and sometimes for animal cell. You have to read the question very carefully. So first of all, we go into the concept of what is cytokinesis. Okay, so cytokinesis is the process of division of what? Division of cytoplasm. I think you all know about it, right? And this process happens after karyokinesis. That is the nuclear division. So first is the nuclear division, then it's the cytoplasmic division, and they both are the part of cell division. Okay, and the process of cytokinesis in the plant cell and the animal cell is different. So first of all, starting with the animal cell. So in animal cell, cytokinesis, it proceeds by furrowing and invagination. So it proceeds by furrowing and invagination. Got it? Furrowing and invagination of plasma membrane at the plane of the equator, at the plane of the spindle equator, of the spindle equator. But in case of plant cells, in case of plant cells, what happen, that here the cell wall is present. Okay, so the furrowing process is not possible here. So here what will happen, the cytokinesis is assisted by the formation of phragmoplast and cell plate. Okay, cell plate in case of plant cells, because furrowing here in case of plant cells is not possible because of the presence of the rigid cell wall. Okay, because of cell wall, I'm just writing in short. Okay, so if we elaborate about animal cell a little more, how it is, let's see. So the key determinant of the plane of cytokinesis in the animal cell, now we got to know, is the central spindle. So if we describe about animal cell here, the key determinant is what? Yes, tell me, the key determinant of the plane of cytokinesis is central spindle. Okay, because in plant cell it is cell plate and phragmoplast, is central spindle. Now there are several spindle fibers which are attached to the spindle apparatus, right? If you know that the spindle, if these are the spindle fibers which will hold the chromosome. Okay, so these spindle fibers are the part of the spindle apparatus. Okay, and this spindle fibers, they help in chromosomal segregation, moving the chromosome to two opposite poles, right? They are all the part of spindle apparatus, and they help in chromosomal segregation during both mitotic and meiotic cell divisions. Now these spindle fibers, if we elaborate, the spindle fibers are of three types. What are those? Number one is chromosomal spindles. Chromosomal spindles. Also, they are called as kinetochore microtubule. Kinetochore microtubule. Why? Because these spindle fibers, they are attached to the kinetochore. They are attached to the kinetochore. That is why they are said to be as kinetochore microtubule. Okay, what they do, they will pull the chromosome towards the pole. Okay, they'll pull the chromosome towards the pole. What they will do, they will pull the chromosome towards the pole. Okay, so that there will be the proper segregation. Next is astral fiber. These are said to be as astral microtubule. First is kinetochore microtubule, second is astral microtubule, and what is third? First let me write the name. The third is central spindle. Okay, it is said to be as polar microtubule. Polar microtubule. Okay, so astral fibers, these are not attached to any chromosomes. They simply radiate from the centromere. Radiate from centromere and not attached to any chromosome. And the last one, this last one, these are the microtubules. They emerge from the opposite pole. They emerge from the opposite poles and they are overlapping. Okay, they emerge from the opposite poles. I'll show you with the picture. Opposite poles and they are overlapping. Okay, and these central spindles, they are the key determinant. The central spindles are the key determinant of the plane of cytokinesis in case of animal cells. And these central spindles, they recruit several proteins to the central portion of the cell, as a result of which this central spindle will help in furrowing of the plasma membrane. Okay, so furrowing of the plasma membrane will take place by the recruitment of several proteins onto the central spindle. Okay, now, so if I draw with the three types of spindle, how it will look like. This is the chromosome and toward the centromere is the kinetochore. So I'm drawing two centromeres and the kinetochores which are near the centromere. So the towards the two ends are the astral fibers. Okay, are the astral fibers. These astral fibers are going to form astral microtubules, and the ones which are going towards the pole, and these are overlapping. Okay, and there are some which touches the kinetochore. Okay, so the one which touches the kinetochore, what was that, guys? Kinetochore microtubule. Okay, and the ones which are overlapping at the end, what are those? What are those? Yes, these are the polar microtubules. Polar microtubules. These are the kinetochore microtubules. Okay, so these are the astral microtubules, which is forming astral fibers, and this is the kinetochore microtubule. I am marking it with the another color as well. So this is the kinetochore microtubule, this one. Okay, this is the polar microtubule, this one, and these are the astral microtubule, the one which is radiating from the this region. Got it? So what will be our answer? So the key determinant of the plane of cytokinesis in mammalian cells, it will be what? It will be central spindle, because this is towards the outer side and this is going to form what? It is going to form the furrow from the plasma membrane. This is the key determinant, and this is also said to be a central spindle. Moving to the next question. Beating of cilia is regulated by. So let's first understand about the cilia. So eukaryotic cilia, it shows 9 plus 2 arrangement, right? We all know eukaryotic cilia have 9 plus 2 organization and is composed of 9 outer units and 2 central units. Okay, and the 9 outer units, it is composed of one complete unit called A and one incomplete unit called B tube. Okay, so I just explain you here. Cilial axoneme, it shows what kind of arrangement? 9 plus 2 organization. And in this 9 plus 2 organization, the 9 is the outer unit and this 2 is the central unit. Got it? Now, the 9 units, out of this 9 units, 9 outer unit, this 9 outer unit is composed of one complete unit, which is called as A unit. I don't put the arrow here, which is called the A unit, and one incomplete unit, which is called the B unit. Got it? Or the B tube. Instead of unit, we say it as tube. Instead of unit, we'll say it is a tube. And this central unit is composed of single complete unit. Only complete units are there in the central unit. Okay, now coming to the another part, that is dynein. Now this dynein arms, there are dynein arms which are seen as projections, fussy projections from the A tube. Okay, I change the color, that these dynein parts are the fussy projections from the A tube. Okay, and these dynein arms, they are of two types. These dynein arms, they are of two types. What are the two types? The one is outer three-headed. This is outer and another is inner. Okay, the outer is three-headed and the inner is two-headed. Okay, and this 9 complete unit, okay, this 9 complete unit, this is towards the periphery. This 9 is towards the outer unit. We say it as they are in the periphery. And these peripheral units, they are interconnected to each other. Okay, they are interconnected to each other by nexin, a protein named as nexin. Okay, so this 9 outer unit, they are interconnected by nexin, by a nexin bridge. Okay, and what is the role of this nexin? What is the role of this nexin? This nexin regulates the beating of the cilia. Beating of the cilia. And where does this energy for the movement comes from? They come from the dynein arms. What these dynein arms do? These dynein arms, they are the motor proteins. These dynein arms, they are the motor proteins. What do? They convert the chemical energy from ATP into mechanical energy for movement. Understood? So moving to our question, beating of cilia is regulated by nexin and the energy is provided by the motor proteins dynein, which converts the chemical energy of ATP into the mechanical energy. Moving forward, 28. Cystic fibrosis transmembrane conductance regulator, CFTR, in short it is said to be a CFTR. It is known to control the transport of which iron? So let's first understand about this CFTR protein. So this CFTR, this is what it is. This is a transmembrane conductance regulator. It is a transmembrane protein. It is actually a transmembrane transporter. And what does this transport? What does this transports? It transports chloride, thiocyanate across plasma membrane. Okay, across plasma membrane of epithelial cells. And these CFTR, they belong to a class of proteins known as ABC transporter. They are ABC transporters. What are ABC transporters? They are ATP binding cassette transporter. ATP binding cassette transporter. Understood? And these ABC transporters, they are kind of ion channel protein category. This ABC transporters, what they are? They are ion channels transporters. Got it? Now, this CFTR, it is very important transporter because it helps in the maintenance of chloride ion channel functioning. Okay, and if this CFTR is damaged in the cells, in that case it will help in causing problem in the fluid transport. Okay, so what happens if this CFTR is impaired? In that case, what will happen, since it is involved in chloride transport, thiocyanate transport? In that case, what will happen? There will be problem in epithelial fluid transport, especially chloride ions. And this CFTR, when this is impaired, it causes a disease known as cystic fibrosis. Cystic fibrosis. And this cystic fibrosis, it is a autosomal genetic disorder, recessive, autosomal recessive disorder in humans. Okay, and whenever this CFTR gene, since a recessive disorder, that means both the genes of CFTR, both the genes which encode CFTR has to be mutated to cause this disease. Okay, so it is autosomal recessive disorder in humans and it is caused by impairment or the mutation in the CFTR gene. Okay, so I write here caused by mutation in CFTR gene. Now moving to the question. Cystic fibrosis transmembrane conductance regulator, CFTR, is known to control the transport of which ion? It is chloride. Answer 4. So this was all about the cell biology. I hope this part is clear. We'll move forward with the unit 3 in the next video. Thank you so much. Please like and subscribe the channel for more videos. Your feedback and comments are highly appreciated. It will help me to improve my videos. And also, if I can help you in any way, please write it in the comment box. Thank you so much, guys. See you again.